Welcome, folks. Thank you so much for coming this evening to the third in our series of talks about what's new in the fall of the Roman Empire. A lot. Sponsored by the Initiative for the Science of the Human Past with the support of the Department of Classics and the Department of History here at Harvard. The initiative, our thing would like to work. Oh, this is getting interesting. What's new in the fall of the Roman Empire? Yay. The initiative of the science, the Initiative for the Science of the Human Past at Harvard aims to bring together the power of the advanced sciences and the questions and inspirations of the humanities and the social sciences to illuminate the human past in all its splendiferous complexity in which our present is so deeply embedded. Tonight is a very, very special occasion that you've happened on mostly unwittingly in the life of our initiative. For this presentation marks the first public beginning of an exciting and amazing new transatlantic collaboration, a joint venture of the SoHP, the Initiative for the Science of the Human Past, with the new Max Planck Institute for the Science of Human History in Llena, whose founding director, Professor Johannes Krause, will be speaking to us this evening. Our new research program here and there will be called, on both sides of the Atlantic, the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean, which I suspect we'll pronounce as MHAM. Now, if that name sounds like a bunch, like it might have come out of a committee, I can tell you that it did come out of a committee, and the very long tractations of the Office of General Counsel of Harvard University and that of the Max Planck Society in Munich. Anyhow, it's working, it's great, this is the beginning. Our two previous talks in this series were brought to us by Kyle Harper in October, who memorably addressed questions of infectious disease and ecology. Nature did it, was his, in some summary answer to the fall of the Roman Empire. And another talk in December on new ways of studying the fall of the Roman Empire in the 21st century. Tonight we're going to zoom out time-wise and zoom in focus-wise with a talk by a remarkable scientist. One would be tempted to call him a rising star if it were not true that his star has already risen swiftly and spectacularly. Johannes Krause has been doing amazing things in very short order. And those amazing things are contributing in a very important way to the questions that historians and archaeologists are wrestling with right now about the fall of the Roman Empire. Starting with this question, did mass death from disease knock the wheels off the Roman Empire's economy and usher in a final collapse of the Roman imperial system that had dominated the Mediterranean, its civilization, its economy, its geopolitics, for some 700 years by the time of the reign of the Emperor Justinian in the 500s. Although some have controverted this, new evidence is fast accumulating beyond the written sources we have long known. For instance, about the nearly 250,000 deaths recorded in the imperial capital at the first outbreak before the imperial accountants gave up counting in despair. The new evidence is deepening and changing our understanding of the terrible pandemic that struck the Mediterranean and beyond in awful and successive waves from 541 to about 750 AD. Archaeological evidence is piling up in the form of mass graves recently inventoried and mapped. Biomolecular evidence is piling up also in the form of the robust identification from ancient DNA of the pathogen itself. There is now no doubt that the new disease, the main killer that stunned the late Roman Empire in the 500s and subsequently was Yersinia pestis, bubonic plague, now also identified as the pathogen behind the medieval Black Death. In this astonishing voyage of biomolecular discovery of ancient pathogens and of their evolutionary and epidemiological trajectories, Professor Krause and his group have played a truly leading role. As a child growing up in Thuringia, then for still a few more years in East Germany, the young Johannes, as he has told me, always loved history. But that did not seem like such a smart career option in those years, unlike today. And he became a biologist. He later earned his PhD at the University of Leipzig and worked extensively with Svante Pääbo at the Max Planck Institute for Evolutionary Evolutionary Anthropology, also in Leipzig. Then he began a spectacularly swift rise that had him go from assistant professor to full professor of scientific archaeology at Tübingen, where we first met, right in the lab where DNA was first identified in the 19th century. And then, in another instant, it seems to me, he became the founding director of this magnificent new Max Planck Institute for the Science of Human History in Llena, which is bringing unmatched resources and amazing talent and intellect to exactly the same task that Science of the Human Past has been pursuing here at Harvard at a more sedate rhythm in its present form since 2011. From Professor Krause's long-standing and fruitful collaboration with David Reich in his lab, and from our own friendship and collaboration, has emerged the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean, MHAM. My own group of graduate students has profited immensely from their collaboration with Professor Krause's group and his students in combining the historical written evidence, the philological analysis of the Greek, Latin, Arabic, Syriac texts, and the archaeological and biomolecular archaeological evidence, genomic evidence, for reconstructing the course and nature of the various waves of bubonic pandemic at the end of antiquity. Johannes has made wonderful contributions to archaeogenetics. It would take me all night to go through them in detail. From the question of the FOXPRO gene among Neanderthals, to identifying the pathogen, something dear to Bostonians, the pathogen, or not so dear, behind the Irish potato blight and famine. About our subject tonight, Professor Krause's group made a fundamental contribution with that of Henrik Poinar when they were able to reconstruct a draft genome of the bacterium of the Black Death in 2011. Since then, Professor Krause has made a series of signal contributions, including reconstructing the details of the Bubonic plague genomes that struck Marseille early in the 18th century. Most recently, his group has recovered a high-coverage Yersinia pestis genome from a 6th century Justinianic plague victim in Bavaria, only the second site to yield robustly authenticated evidence of the Justinianic pandemic's pathogen. We are now, in other words, entering a new biomedical and archaeological world in which collaborative ventures are illuminating the deep evolutionary history of disease in exquisite detail, changing our understanding of the nature, extent, impact, and course of ancient and modern pandemics, with abundant lessons for specialists of modern infectious diseases, as well as for archaeologists and historians. Tonight, Professor Krause will take us on a remarkable tour of the longue durée of the terrible disease of Yersinia pestis, which, to our astonishment, we learned only a year ago already existed in the Stone Age. He will follow that story down to the form that struck France in the 1720s. And to spur our thoughts and discussion in the time that will follow, we will have the privilege of hearing brief comments on the talk from the historical perspective from Professor Kyle Harper of the University of Oklahoma, and from the microbiological perspective from Tom Benjamin, member of the Steering Committee of the Science and the Human Past, and with whom both I and Professor Harper got our first experience with ancient DNA, trying to identify that of Yersinia pestis in vain in his lab in the department then of pathology at the Harvard Medical School. But with no further ado, please join me in welcoming Johannes Krause of the Max Planck Institute for the Science of Human History and of MHAM, who will speak to us tonight on the genetic history of plague from the Stone Age to the 18th century via the Roman Empire. Johannes, welcome. This isn't working for moving the slides.
Okay, remote control doesn't work, but here. So first of all, thank you, Mike, very much for the invitation, and it's my pleasure to be here and talk today about some of the research that my group has been focusing on over the last few years and trying to uncover the genetic history of Yersinia pestis, and it has actually brought us from the medieval time down to the Roman Empire and now even into the Stone Age, and I hope I can take you on that voyage also tonight. And it's also my pleasure to be here and kind of starting this initiative that Mike has already mentioned in his presentation, this new Max Planck Research Center together with Harvard and the Max Planck Institute in Llena, which I'm very excited about, which I would probably pronounce MAM and not MHAM, but I'm certainly open to any kind of suggestion on English pronunciation. Like I mentioned before, this is really a voyage that has brought us into many different time periods now. The Roman Empire is just one slight, small chapter, I would say. I think most of it is probably in the medieval time of what we know, certainly about the genetic history of the plague currently. But as I said, and as Mike already mentioned, it even brings us now into the Stone Age. The history of most infectious diseases that we find in the human population today actually starts probably even earlier, probably sometime in the so-called Neolithic. Noticing that the presentation is again a bit smaller. I think we have actually found how to make it bigger, so I will try that now. Okay, no, that doesn't really work, so let's keep it. Work. Uh-oh. Doesn't look good. I just see the pizza of death. But maybe it's a bit.
But it's clear.
It's okay. The writing is a bit. Now I can already see on the next slide that has completely jumbled the text. So I better leave it a bit smaller. So people that don't see in the back should come into the front. There's a wonderful red couch here. Looks like Sigmund Freud's, but. Yeah, that seems to work better. So it is really a story that probably started already pretty early on in human history. Some people suggest that it was sometime in the early Neolithic, so the time periods when human became sedentary, farmers, agriculturalists, they started to live in much denser quarters together and actually provided then very good conditions for pathogens to spread. And it is also hypothesized that many of the pathogens that we see within the human population today were actually transmission from our domestic animals that were domesticated during that time in so-called zoonotic transmission, because we find the closest relative of those human diseases, such as measles, smallpox, flu, tuberculosis, plague, leprosy, pertussis, actually today in our domestic animals. So this is why it's thought that many of them actually entered the human population from those animals that can be domestic animals, but also commensals that live in close proximity to humans, such as, for example, mice or rats. And then, of course, when human populations became even bigger, civilizations started, some of those pathogens then caused major outbreaks that we then also know from historical records. Some I mention here, like the Plague of Athens, two that struck the Roman Empire, so the Antonine Plague and the Justinian Ik Plague, and then, of course, the infamous Black Death, this major pandemic that happened in the 14th century, which is probably something that most people are quite familiar with. What was very unclear from the historical records, however, was what pathogens were actually responsible for those large outbreaks in the past. I put some question marks under smallpox or measles for Plague of Athens or Antonine plague, but for the two later ones, the Justinianic plague and the Black Death, I will actually show you we have now very strong molecular evidence, as Mike already mentioned, that those have probably been caused by Yersinia pestis or bubonic plague. Still, for many people, if you kind of hear about plague and smallpox, that sounds more like something from the past, something that is actually not so relevant maybe to us today. It is relevant for historians, it's relevant to understand what happened in the past, but it might actually not be so relevant, say, for medical research, for example. And in fact, if we go back, say, 60, 70 years, there was quite a lot of medical researchers that said even infectious diseases are not so interesting anymore to be researched because we have vaccination programs, because we have antibiotics, so that by the end of the 20th century, people actually suggested there will be no infectious diseases anymore because of this amazing kind of new medical treatments that we had. But of course, we know today that this unfortunately did not happen. In fact, there's only a single pathogen that has been more or less eradicated, that is smallpox, even though it's not completely eradicated because there's still two labs in the world, one in Atlanta and one in Novosibirsk, that still basically have this pathogen in case somebody might use it as a bioweapon. They could still do research on it. But what we also see is that since the 1980s, we see an increasing amount of new pathogens emerging from different parts of the world. Of course, everybody has seen and heard much about HIV, but also SARS, Hanta, the Ebola outbreak from only 2 years ago in Western Africa, are all examples of pathogens that are spreading in the world today. So infectious diseases are still very much a topic in medical research. And the other problem we have is re-emerging infectious diseases. Some of them become antibiotic resistant, like, for example, tuberculosis, which is actually spreading in the world today again, causing thousands, and even in case of tuberculosis, 100,000 of casualties every year. So this is a very, very hot topic of research. However, there's actually very little that we know about the early evolution of pathogens. And that is mostly due to the fact that if you want to study evolution, in most cases, people have fossils to look at. So I've been working a lot in the past with Neanderthals, I've been also working with mammoths and cave bears and all kind of megafauna. And for all of them, we had a pretty good understanding already about their evolution, because we can actually look at bones, fossils from the past. We could actually understand how they changed through time. However, for pathogens, we often, of course, do not have that. We don't have fossilized bacteria. Even if we have them, they're just a round shape, but they don't really tell us much about the biology of that round shape. Right? There could be any type of bacteria that we are looking at there. So therefore, we know very little about the early evolution of the pathogens, about evolutionary rates. So how fast do pathogens change over time? What's their mutation rate over hundreds of thousands of years? And also we know very little about host-pathogen interaction. So how do the pathogens change to become human pathogens, but also how do humans react through time to the pathogen? Are there actually genetic changes in the human population? For example, resistancy genes that are spreading within the population. That is something that has really not been much investigated and understood. And this is basically what we noticed about 5-6 years ago, that there's really a lack of information from the past to study ancient pathogens. And this is when we started this new field that we now call ancient pathogen genomics, which is basically researching directly ancient pathogens by taking skeletons from the past and reconstructing the genome of the pathogens that might still be left behind in those ancient skeletal remains. For example, taking teeth, taking bones, taking mummified tissue from the past and extracting DNA from that to be then able to tell something about the evolution of those pathogens. And that basically provides some sort of molecular fossil record. We don't get kind of fossils in the kind of form of some sort of shape or bone, but we basically get a molecular fossil, a genome that we can then analyze and compare to modern pathogen genomes. We learn then more about mutation rates through time. We learn something about those host-pathogen interaction. And, of course, we can also provide historians, provide archaeologists with this important information of what actually caused those millions of casualties in the past. So like the Justinian Ik Plague, the Black Death, the Antonine plague, and so forth. So what pathogen was actually responsible for those large pandemics in the past? And we've done quite a number of research projects together with my now 3 group leaders, and back in the day postdocs and PhD students, Kirsty Boss, Alexander Herwig, and Verena Schünemann. And the first where we started was the Black Death, something I will also focus today a bit about. But we also looked at ancient leprosy, so we were actually able to reconstruct leprosy genomes from medieval and now also early medieval time. As Mike already mentioned, we've also been able to reconstruct the causative agent of the Irish potato famine, which is actually not a human pathogen, but it's a plant pathogen that infected potatoes at the time. And we've also looked at Mycobacterium tuberculosis, in fact, from the New World, pre-Columbus, which was very exciting because people thought it was actually not there pre-Columbus, but we could show it was there, but it was not a human form of tuberculosis, but in fact a seal tuberculosis, which was very exciting. So some zoonotic form that was transmitted from seals into human populations. And then recently we worked on the stomach contact of this fellow here, which is the Iceman, the Ötzi, an ice mummy that was preserved in the Alps for more than 5,000 years, and we could actually reconstruct from the stomach content Heliobacter pylori, so bacteria that live in the stomach that can cause ulcer, that can cause also stomach cancer, and it was quite interesting to compare this 5,000-year-old bacteria to modern bacteria and see how they changed through the last few thousand years. But then today my main focus is really plague. So what we know about plague today is that Yersinia pestis is usually not found in human populations. It's actually not a human pathogen per se. If it is found in human populations, it's usually some sort of accident. Right? Usually it's found in wild rodent populations, such as squirrels, germals, marmots, and eventually it can get transmitted from those wild rodent populations into commensal rodent populations that live in close proximity to humans, for example, rats or mice. And the transmission happens actually by fleas. The fleas kind of bite infected individuals, and they take the bacteria up in their stomach. And what happens in the stomach of the flea is actually that those bacteria, they build a biofilm that clogs the stomach of the flea. So the flea can actually not swallow the blood. What then actually happens is the flea is actually starving, and it keeps on biting. And every time it bites, it actually kind of injects bacteria into the bitten individual. What then happens to rats and mice is that they actually also get bubonic plague and they die. And if they're dead, the fleas look for the next warm body, and if that is not a rat or a mice but a human, then actually humans can get infected, and that can then cause bubonic plague. So the bacteria kind of migrate to the next lymphatic node, somewhere close by. They get swollen, the bacteria replicate here, you get this bubo, which then gives the name bubonic plague. And then the bacteria basically replicate, they move into the body, and if you don't get antibiotic treatment, in about 50% of all cases, after 7 to 10 days, it causes death. It actually causes multiple organ failure. You have necrotic tissues, so hands and feet often get necrotic, they turn black. This is where the name Black Death came from. And it's quite high mortality, so 50% that is comparable, for example, to Ebola. But this is actually not the only form how people can get infected. There's also a form where humans can infect other humans, not by direct flea transmission, but actually by oral droplet infection. If humans can get infected, the bacteria move through the body and then infect the whole basically body. They eventually also infect the lungs. And if you have an infected lung and you cough up bacteria, somebody else inhales the bacteria, you can get something that we call pneumonic plague, which is actually a very severe form of plague, because when you see the first symptoms, which is usually after 15 to 20 hours, you only have about 5 to 10 hours to live, because it's very, very fast. So mortality is still today very high if you get infected directly into the lung. Antibiotic treatment is usually too late because the symptoms arise so late, and within 24 to 48 hours, people actually die. And there has been actually even a few cases where people got infected in the lab, for example, by inhaling the bacteria. It's also in the Grand Canyon a case a couple of years ago where somebody was dissecting a mountain lion, and probably that died of the plague itself, the mountain lion, and doing the dissection probably inhaled the bacteria, and the next day the person was dead. So that's pretty bad. So Yersinia pestis is still found in the world today. There's actually many places in the world where it's found. Most common, it is found in South Asia and East Asia, but there's also some reservoir population in Madagascar where there's outbreaks every couple of years. And then also in the United States, especially in the western part of the United States, there are reservoir population, mostly squirrels or groundhogs, that actually carry the bacteria. And again, there's a few dozen cases in the US and a few thousand cases worldwide where people can or get infected from the bacteria and kind of have bubonic plague or some pneumonic plague form. But usually mortality today is quite high because people know how to treat it. There's a number of antibiotics that can actually be used for treatment, and they are actually quite efficient. But if you don't do basically any antibiotic treatment, then mortality, of course, is still very high. So the pathogen has kept its virulence. It hasn't really lost it through time. So if you then look back into the past, trying to understand ancient pandemics and trying to see which ones might have been caused by the plague, the first one that was already mentioned by Mike, where people have suggested, based on historical records, in this case it was Procopius, who basically reported an historical text about this outbreak that we now call the Justinian Ik Plague, which started in the mid-6th century, based on the very good reports that she actually gave about this disease. People suspected that it might have been caused by Yersinia pestis, so it was a plague pandemic. This is also why it was called then already some years ago the first plague pandemic, even though no molecular evidence was available, but the historical records really suggested that Yersinia pestis caused this outbreak. The second pandemic then that people thought was probably related to Yersinia pestis outbreak was the Black Death, which happened in the mid-14th century. It was a very short time period of only about 5 years. It is thought today that about 50% of the European population died in this major pandemic. It then stayed on in Europe. There are then multiple outbreaks in the next 350 years that happen again and again in waves. It's about 18 to 20 waves of plague, where we'll talk later about if they have actually been independent wave or whether it was the same pathogen that caused it. And then the last big pandemic that we think was caused by Yersinia pestis was in the 19th century. It actually started in the Yunnan province in China in mid-19th century. And some people say it's still ongoing. Other people say it kind of disappeared and was antibiotics in the mid-20th century. But it was basically around for about 100 years. And again, it killed millions of people, especially in East Asia. But through the harbors of Hong Kong, it then also disseminated the disease all over the world. So the fact that we have it in the western part of the United States today, that we have it in Madagascar and in Central Africa, is probably due to this event here, that basically by the ship traffic, by the globalized world, the pathogen actually spread by ship traffic all over the world. So what we have then been interested in when we started this work is trying to see and trying to identify which of those larger outbreaks were actually caused by the pathogen, because when we started this work, there was a big debate among historians whether especially those ancient historical pandemics were caused by Yersinia pestis or whether they might have been actually caused by some other pathogen. In fact, some people suggested, especially for the Black Death, there was some virus, some hemorrhagic fever, a pathogen that cannot be identified anymore because it's extinct. There's been really a lot of debate, and there's been some early molecular work that was disputed by other ones that suggested that it was all contamination. So it was a really hot debate that we basically stepped into. But the molecular identification of plague is actually not so straightforward. It's actually not so easy. Because if you have ancient skeletons, and you look for molecular evidence, you look for genetic material like DNA, If you extract DNA from an ancient skeleton, like an ancient tooth or bone, you actually get a soup of DNA, all kinds of DNA. That is the DNA of the human itself, but actually the vast majority of the DNA that you get from ancient skeletons is not even from that human. It's also not the bacteria that this person carried, but it's actually environmental DNA. Because a bone is really like a sponge. It basically sucks up all the DNA from all kinds of microorganisms that live in the soil. So you really get an environmental sample if you want. So trying to identify then the pathogen from the soup of DNA is actually not very easy. The one possibility is what we call a shotgun approach. So we have this wonderful sequencing technologies today that can produce millions and even billions of DNA sequences. So you can just take an ancient sample, extract the DNA, and sequence the hell out of it. If we do that, however, we're stuck with the problem that now you have millions of DNA sequences that you have to try to sort, to try to identify, to try to know which DNA sequence actually comes from which organism, and which DNA sequence actually might be, for example, from the ancient pathogen. We have been working hard in the last few years on tools to identify those DNA sequences. So this is a field that we then call metagenomics. So you take DNA from environmental samples and trying to make sense which DNA fragment comes actually from which organism. And one of my group leaders here in the department, Alexander Herbig, has developed this tool that we call MOLD. I don't want to go much into detail here, but what this tool actually is an alignment tool. So it takes all those DNA sequences and compares it to all genomes that have been produced in databases. And that is something that people have been doing already for 20 or 30 years, and with a tool that's, for example, called BLAST. But that tool is very slow, whereas this tool is much, much faster. It's several hundredfold faster than kind of the normal tool that people have been using in the past. So it is so fast that we can now actually analyze 1 billion DNA molecules in less than 24 hours. So we can really produce massive amounts of DNA sequences from those ancient samples, and then just say from which organism every DNA fragment comes from. Which is great, but again it produces a problem. And the problem is the output is very, very difficult to read, because the output basically looks like that. So you see this phylogenetic tree, hard to see for the people in the back, I guess. But basically, if we take such a sample, we see something like 2,500 different organisms' DNA in such an ancient skeleton. And now it's very tedious to find out which one of that could actually be the pathogen that has been responsible for the death of that person. So you can then zoom in, and then you look, for example, in this case, we have a lot of mycobacteria that cause tuberculosis, but you see actually many, many, many different species of mycobacteria, hundreds of different mycobacteria. So which ones are now actually the pathogenic ones? Which one are now actually the ancient pathogen? Which one might just be from the soil? So it's quite difficult. What we can then do is we can look at each individual DNA fragment, compare that to a reference sequence here. So this is a sample where we suspected it might have tuberculosis. You have many, many DNA reads. You can actually, if you compare that to the reference, see that they have a lot of differences. So every kind of colorful bar here is actually a difference to the reference. You can actually see it's less than 90% identity. So this is something we would call a false positive. So this is probably not TB. Whereas in this sample here, we have a lot of reads. They're looking very, very similar. So more than 99% sequence identity. In this case, we would say that's a true positive. That's indeed tuberculosis. It's not quite easy to find then the pathogen, especially if you don't know what pathogen might have been present in this ancient skeleton. Sometimes we're in a much better situation, like for example, for this particular cemetery here that we have been first studying about 6 years ago, where we got skeletons from the East Smithfield Cemetery, which is in the central London, and those skeletons here were excavated in the 1980s when they were building one of those beautiful cucumber-like new office buildings close to the Tower of London. So there's the Tower of London here. They found about 500 skeletons, and based on historical records, they knew that this was a plague cemetery. The historical records actually said that it was only used in the Black Death because they had so many dead people in the city of London that they turned this field into a cemetery and they closed it afterwards again. So So we can actually be rather sure that most of the skeletons that we have from this site died during the Black Death. So now we have some idea, okay, that could then be Yersinia pestis, because that's something that people have been discussing was responsible for this particular outbreak. So what we then did is we analyzed 50 skeletons from this collection, and indeed in 20 of them we could find with classical PCR Yersinia pestis. At this point, we could have actually stopped our research. At this point, we could have said, okay, Yersinia pestis is present. We can show Yersinia pestis was present during the Black Death. It was likely the causative agent of the Black Death. But we actually wanted to go one step further. We actually wanted to go one step further and reconstruct the entire genome of this bacteria to understand better how it actually changed through time, what we can actually say about the genomic elements of this ancient pathogen. But that is now very difficult because, again, if we look at the DNA that we get from the skeleton, there's such a tiny fraction that is actually Yersinia pestis. How do we get enough pieces to puzzle together an entire genome? And to do that, we have developed methods that we call DNA capture, where we have devices such as, for example, those microarrays, which are small pieces of glass. And on the surface of this piece of glass, you actually synthesize DNA. You can, for example, synthesize an entire genome of Yersinia pestis, of modern Yersinia pestis, and then incubate your DNA with this piece of glass. The fragments that are similar to the DNA on the piece of glass will actually bind to this piece of glass, and everything else that is not similar you can actually wash away. By that you enrich for Yersinia pestis DNA, you can put it in one of those wonderful sequencing machines, and then reconstruct the Yersinia pestis genome based on those hundreds or thousands or millions of pieces. And this works actually quite well. If you see the amount of Yersinia pestis DNA we got from the skeleton before we did enrichment, you can see it's very, very low, and after enrichment it was actually very, very high. You then still have another problem, and that is the problem that if you then have that DNA from this ancient skeleton, some people might still say, how can you make sure that this is indeed 660-year-old DNA? Couldn't that also be bacteria that just live in the soil today? Couldn't that be something that is actually modern contamination from the lab? So how can you actually show that this DNA is indeed old, that it's authentic ancient DNA? And to do that, we have developed some methods in the last couple of years where we look at some specific properties of DNA when it gets old. Because when DNA gets old, like everything, it actually falls apart. And actually, DNA accumulates something that we call DNA damage. And this DNA damage, so chemical modifications to the DNA, cause changes to the DNA sequence, specifically changes where C's in the DNA become T's. And that leaves a very characteristic pattern. So if you look along the DNA fragment for those changes, you can actually see that there's a lot of changes at the beginning of the DNA fragment and at the end of the DNA fragment. So basically this damage accumulates in this part of the DNA that is not so well protected like the inner part of the DNA. And that is something that we see in Neanderthal DNA, which is 40,000 years old. We see that in DNA from any kind of megafauna from the past. But where we do not see that is in contamination, and we don't see that in DNA from modern people or from modern DNA from the lab. We could even show that it accumulates through time. So this is time, this is the amount of damage. So the older the DNA gets, the more damage accumulates. So if you have, for example, samples that are about 100 years old, they have about 10% damage. If you have some that are 500 to 2,000 years old, they have about 20% damage. So if you then look at the DNA from, for example, the Black Death time, we look at the human DNA, it has this typical smiley here, so it has a lot of damage. Whereas then if you look at the Yersinia pestis DNA, we can actually see the same pattern. So therefore we can assume, so we see 20% damage, that this DNA is indeed about 500 years old, right? But that really tells us that this DNA must be old. That cannot be modern contamination. It cannot be something from the soil. This is actually ancient DNA that is hundreds or even thousands of years old. And that basically then allows us to reconstruct really complete genomes, the complete blueprint, basically, of this ancient bacteria from the past. So in this first study, we reconstruct about 99% of the genome of this ancient bacteria. We have seen every position about 30 times. So we have a very high quality sequence that is really comparable to modern bacterial sequences that you could produce in any microbiology lab. What we can then do, if we have those ancient genomes, we can actually build phylogenies. We can build family trees of ancient and modern bacteria. And those family trees then look, for example, like this. This is the family tree of Yersinia pestis. This is all based on modern genomes of Yersinia pestis. So if you put them all together, those modern genomes, mostly coming from rodents, not from humans, you see that there's these 4 major branches, which we call branch 1, 2, 3, 4, where 80% of all modern Yersinia pestis fall in. Then you have some that kind of fall basal to those ones here, which are called branch 0. And the most recent common ancestor, all those genomes have about 5,000 years ago, which we were able to calculate using the ancient genomes, as you will see. The closest relative is actually a soil bacterium. That's something that is pathogenic to people, but it's not getting transmitted by fleas. If you would get it into your blood, it would still kill you, but it cannot be transmitted by fleas. When we then take this family tree of modern Yersinia pestis, we can then take our ancient genomes and stick them into this family tree to see how the ancient ones are related to the modern ones, to tell us something about what might have happened in the past and how modern and ancient ones are related. And when we do that for the Black Death, for this ancient genome that we reconstructed from the London site, we can actually see it falls right here in this position, very close to the most recent common ancestor of all those different branches here, which 80% of all the modern strains fall into. That was actually very exciting to us when we saw that the first time. Because you can actually already imagine, if you just look at the family tree without having this ancient London strain here, that something has happened here. Because you have all those strains diverging like a star from each other. And that's usually the sign of a pandemic or some major event. So you basically see that the population size of this bacterium is increasing a lot. So it's basically spreading within the population, and that we seem to pick up here. And it's also very exciting for us to see that it basically falls very close to this position here, so that the Black Death strain seems to be close to this event where the bacteria are spreading, which makes a lot of sense, because the Black Death is a major pandemic, so this is probably also then seen in this tree here. But then that was not enough to us, because we still wanted to understand more about what actually happened after the Black Death and during the Black Death, because there were many different hypotheses that people have put out on this ancient pandemic. And one hypothesis was whether there were multiple introductions of plague into Europe. And that is true for the Justinian Ik Plague, that is also true for the Black Death. Because we have different strains, we have different events, we have 20 different outbreaks after the Black Death in Europe. Was it every time brought into Europe, again, by people, traders, ships, any kind of contact with Central Asia or Eastern Asia? Or was it actually that there was some sort of reservoir population? Was there actually a rodent population in Europe that carried the bacteria and gave the bacteria again and again into the human population? And what we also wanted to look at is whether the bacteria actually traveled all the way back to Asia. Because if you actually paid attention in this tree, you could actually see that it was on a branch, go back here, leading towards this branch 1, which was actually the branch that caused the Hong Kong plague in the 19th century. So we wanted to also ask that question, whether the bacteria actually might have traveled back all the way into Asia. To address those, we have in the last couple of years looked at many different outbreaks, many different epidemics, mass burials that we get from Barcelona, from Eastern Europe and Russia here, from Germany, and from a marketplace in southern Germany when they did a parking a lot. They actually found this 150 skeletons in the middle of town that all died during a very short episode, probably within a week or 2 weeks, in a town that had 3,000 people at the time. So basically a mass burial, as Mike has been also researching for the Justinian Ik Plague. And we then wanted to see how do the plague pathogens look like in all those different contexts. We also did a lot of sequencing and a lot of reconstruction again of those ancient pathogens. And if we then look again how they're related to those modern Yersinia pestis genomes, here's again London from the Black Death. The first one that we were actually quite excited to see, that was coming from Barcelona, also from the time of the Black Death, actually falls in the phylogeny here. Have you seen it? Maybe you have. You see it? It falls directly on the London Black Death. It's actually genetically identical. The Black Death from the Mediterranean, and the genome is 100% identical, right? Which is very exciting and interesting because it tells us that this pathogen that killed those 50 million people has genetically actually not changed much in Europe at the time. It was introduced in Europe, it spread in Europe, but it didn't really change, right? Which is quite interesting. We also now have a genome from an outbreak that happened in London in 1360. And that falls in the phylogeny here, very close to the one from the Black Death. It has 3 extra mutations, which is also not a lot. And what is quite exciting also is we have yet another genome that is actually from Bulgar City, which is in Eastern Europe, that falls again on this branch here. We basically have like pearls on a line, those ancient genomes, leading towards this big branch here, which caused the 19th century pandemic. And that is interesting because we basically have microevolution. We have this pathogen evolving, but that is all in sites in Europe, and then leads towards the branch that then later on causes this big pandemic in China, which suggests that actually the evolution of this pathogen happened in Europe, and that it indeed spread back to China. If we then look at Ellwangen and those other sites in Europe in the kind of centuries after the Black Death, they actually built their own little branch here. So this is Ellwangen, this is several genomes from the last large outbreak in Marseille in the 18th century in Europe, and they built their own little branch here, would, if you want, be branch 5. And that is quite exciting as well because it basically tells us that there hasn't been much new introduction of plague. From the Black Death, basically most of the genome that we then find later in Europe, and we have another dozen now, they all fall on this branch. So we basically have a European-specific plague branch that tells us that the plague came to Europe, but it stayed in Europe. So it was not multiple introduction, but it was just around. There was basically a reservoir population that was established in Europe, maybe some rodent population that we don't know about, because rats are not a good reservoir because they also die of it. So it was much more likely to have some other reservoir population, like in Asia, where you have, for example, gerbils, which are basically immune against the disease. They can carry it, but they don't die of it. Right? So you need something like that to have a reservoir over hundreds of years. But then again, Marseille was the last big outbreak in the 18th century, then it disappears. So the reservoir seems to not be present anymore. So we then basically, for the kind of Black Death and aftermath, we have this current model where we think that in the mid-14th century it comes somehow from Eastern Europe into Europe, the kind of reservoir population gets established, but you also have some microevolution of Yersinia pestis and some spread back into Asia. This branch 1 goes back into Asia and then later on in the 19th century spreads all around the world. So that's the same strain that evolved in London, in Europe, that you now find, for example, in the Grand Canyon, right? Or that you find in Madagascar, in Africa, that basically spread then in the 19th century all over the world. Basically, our family tree now gets enriched by another branch, by this kind of post-Black Death European branch, and then this branch 1 here that basically comes out of it and then spreads all over the world. We talked a lot about the kind of late medieval time and early modernity. Of course, I also want to talk a bit more about the Justinian Ik Plague, which is, of course, a very exciting other historical plague pandemic. Mike already did a great introduction and has actually done fantastic research in trying to identify archaeological evidence for this plague, and together we have been analyzing a number of skeletons from different sites, and we have been looking very, very hard, I would say, since the first time we talked about that, to try to find skeletons with Yersinia pestis from mass graves from all over basically the former Roman Empire. But it was very difficult. We really haven't really found too much, but we're quite excited now that we actually have found some. And those come from 2 sites in southern Germany, one called Altenerding, one called Aschheim. They're very close to Munich, that I guess many of you have heard about, not just for Oktoberfest, but city. And the other site is actually Valencia, which is also quite exciting because that is also at least part of this kind of decomposing Roman Empire at the time in the mid-6th century. But we now have genomes, so complete Yersinia pestis genomes from those 3 different sites, and we can put them now into the family tree, right? So if you look at the family tree again, we see all those different genomes here. This is the Black Death, and this is the aftermath of the Black Death. You would expect the Justinianic plague to not fall into this kind of group here. And in fact, they indeed don't do. If you put them in the family tree, they actually fall here on their own little branch, actually a dead end, like a branch that hasn't left any descendants anywhere in the world. Among all the hundreds of genomes that we now have, there's nothing that basically goes back to the Justinian plague. We're not quite sure why. It seems to disappear, like the post-Black Death European branch also is a dead end. It also disappears in the 18th century. Maybe the same has happened here. This would actually speak for the fact that there was an introduction into Europe. The oldest historical records we have of the Justinianic plague are from Egypt, but then spread through the Mediterranean, was around in Europe for about 250 years, but it probably then disappeared. And indeed, between 750 and the mid-14th century, there seems to be no evidence, no historical evidence at least, so far, for a major outbreak of the plague again. So it seems to have disappeared. Again, why is not clear. Some evidence suggests that, again, we had an establishment of a reservoir population, so there was some rodent in Europe where the bacteria were basically thriving in, but again, we don't know exactly which rodent population that was. And of course, there's also earlier plague outbreaks. I mentioned the Plague of Athens, the Antonine plague in the Roman Empire as well, where, again, Galen, an early historian, has actually told us a lot about the disease. We seem to kind of suggest maybe it's smallpox. Some people also suggest it was measles. Other people say it was the plague. It's kind of unclear. And we have also been looking hard for finding evidence for Yersinia pestis in victims from these earlier outbreaks. So far, we haven't found anything. But, in fact, as Mike already mentioned, people have actually found plague much earlier in time. And they actually, by, I would say, accident or coincidence, started to look in population that lived thousands of years ago in the Stone Age. In a very, very interesting time period that we call the Late Stone Age, which was a time of large genetic changes all over Western Eurasia. So that's a time where, together with David Reich's lab here and other colleagues also in Copenhagen, we did a lot of research in looking at human genomes and trying to understand how the human genetic makeup in this part of the world changed through time. And we found that about 5,000 years ago, there was a major genetic turnover in Europe. In fact, between 70 and 100% of the population of Central Europe at the time got replaced by a population of steppe pastoralists that migrated into Central Europe and all the way into the Altai. In fact, this population here in the Altai is almost impossible to distinguish from this population, very different from the previous population. So you have this major expansion of people that happened during the time. Within a very short time period, maybe 100 to 200 years, which is quite exciting. What we then also see is even a backflow, that after this major migration, there is a backflow of human genes into this region that cultures are then called Syntaxta and Andronable. And we and a lot of other people also here in the room, have been thinking hard about what actually caused this major migration. What is responsible for a large genetic exchange and kind of turnover of human genetic makeup? So what happened? Was there some sort of climatic event? Was there warfare? So what actually happened during this time? And this is where our colleagues from Copenhagen had this wonderful idea to actually look for pathogens in the people from that time period. And to our surprise, they indeed found a pathogen, and that they actually found was Yersinia pestis. They found Yersinia pestis in 2 individuals and some evidence in more individuals, where they were even able to reconstruct complete genomes from. One was from this Afanasyevo, this 5,000-year-old culture in the Altai Mountains, and one was from Andronovo, from the same region, but about 3,500 years old. So that was very exciting to us, because now we thought, okay, maybe that was a time of pandemic already. Maybe that was a time where pathogens were spreading, and maybe they are kind of somehow responsible also for the kind of large genetic turnovers you see in the human population. We then did our homework and looked in our samples that together with David we have been working on in the last few years for Yersinia pestis, and indeed we found it in quite a few of them. And those are now actually coming from Europe. So we have been able to reconstruct now complete Yersinia pestis genomes from Latvia, from Estonia, from southern Germany, also from the Yamnaya population in southern Russia, and one also from Croatia in the Mediterranean. Now this is really exciting, because now we can actually kind of put the whole history and evolution of the pathogen even a few thousand years further back in time. We can put those genomes into the family tree of Yersinia pestis. And actually you see that they're building their own branch inside the phylogeny. They're actually on a branch that is slightly outside of the modern genetic diversity. It falls basal. It branches off earlier. It branches off about 5,500 years ago from the other Yersinia pestis genomes. And there's only about 4 mutations from the most basal one to kind of the most recent common ancestor. So it's very, very close here to this time period. So we can basically assume that the kind of divergence of many of those strains started and happened at that time period. So that's kind of the earliest spread potentially or evolution of the pathogen. What you can, however, also see is that there's many of those little branches that are dead ends, and the whole branch itself is a dead end. So there's no Yersinia pestis in the world today anymore that kind of falls on this major branch here. So all the later ones fall on this branch here and don't seem to be related to this Stone Age branch. What is also interesting is you can actually see these numbers here. The numbers correspond to the age, to the radiocarbon age of those samples. And you actually see this is the oldest, second oldest, third oldest, fourth oldest, fifth oldest, sixth. They actually kind of built little kind of branches here on the tree by time. So it's really kind of an evolution of this pathogen over a 1,000-year time period. And you can then also connect them by arrows. And if you do that, which I know historians and archaeologists are never happy when we geneticists put arrows on maps, but we sometimes still do that. You can actually see this is kind of the first. The oldest one is from this kind of Yamnaya population. Then you have this one here in the Altai, one from Estonia, then Latvia first, then Estonia, then you have Augsburg in southern Germany, and then actually the last one is this Andronovo here genome from, again, the Altai region. And if you look at this little map here, that looks quite similar to this map, right? Which is the movement of the people. So it really seems, it's quite intriguing that basically the pathogen seems to follow a very similar route to what we also see in the human genome, where we see this population, Yamnaya, expanding, and then we seem to even have some flow back into Asia. So again, similar to also what we saw in the medieval time. What is also quite exciting is we can now look at the genome structure. So what you see here is basically the modern Yersinia pestis in gray, and then all those Stone Age genomes. And you can actually look if there are genes missing, and for the chromosome we see almost nothing. There's a little gene missing here, and there's a gene missing there. But then on one of those plasmids, which are extra chromosomes, you can actually see there's this big gap here, right? And this is quite interesting because on this gap is actually a gene, which is called YMT, which is the gene that the bacteria need to survive inside the flea. This is actually not present. So from that we can actually hypothesize that this type of Yersinia pestis was not transmitted by fleas, right? So if you look at the basically 3 different forms that we know, that bubonic plague is unlikely because it misses kind of the genes that are necessary for the flea transmission. Which leaves us then with septicemic plague, which basically means it somehow enters the bloodstream, however it does, not by flea biting, or pneumonic plague. Pneumonic plague, however, today is very, very rare. We don't really have many cases in the 20th century. It doesn't really happen that often. So it's hard to imagine that throughout Western Eurasia, there was a large epidemic in prehistory where it was all transmitted orally by droplet infection. But it's not impossible, and currently we can only speculate what happened here, how it was actually transmitted between people. This afternoon we just had a long conversation with David and his team, and we still really don't have a good understanding of what happened here, how this disease actually spread in prehistorical time periods. So then in summary, what can we say? We can say Yersinia pestis caused pandemics in the past, at least the 3 major ones, the Justinian Ik Plague, the Black Death, and the 3rd pandemic were caused by Yersinia pestis, maybe even earlier ones like the Antonine Plague or the Plague of Athens. We can also say that it established a reservoir population in Europe, at least in the time after the Black Death and probably also after the Justinian Ik Plague. What rodent, we do not know. We can only speculate, and there's many interesting suggestions. Some of you might have also followed the literature on leprosy. There was an amazing paper just a couple of months ago where people now found a reservoir population for leprosy that lives in Europe today, that lives specifically in the United Kingdom, and that is actually red squirrels, which I probably pronounced wrong now, I'm sorry. German is Eichhörnchen, that's also very difficult. But they found leprosy and lepromatosis, the closest relative of leprosy, in squirrel populations all over the United Kingdom. Which is actually very exciting because we didn't even know that there's a wild reservoir population of leprosy. We thought it's just a human disease. So that's very, very exciting. We then have a European origin of this lineage 1, which is the major lineage you find, for example, in the Grand Canyon today. So basically the squirrels in the Grand Canyon that carry Yersinia pestis today have a Yersinia pestis strain that once was in Europe and through Asia get transmitted all the way into the west coast of the United States and made its way into the Grand Canyon. So it's quite a voyage that this pathogen actually encountered. And then caused basically this 3rd pandemic in the 19th century. So we really see a high mobility of Yersinia pestis throughout human history, throughout the last 5,000 years at least. And that is interesting because it's a rodent-borne disease. So you would really have to move the rodents with the pathogen, which is kind of hard to imagine. It certainly makes sense as soon as you have ships, and you basically have ships as a vessel to kind of bring the disease somewhere. This is how it arrived in California. This is probably also how it arrived in the Mediterranean during the Black Death. But at least in prehistorical times, it's kind of hard to imagine if people really carried rats when they were on the horses as pastoralists in the steppe. It's kind of really hard to imagine. And then, of course, there's all kind of possibilities. This mobility in humans might, like trade, migration, globalization, cause them basically the dissemination of the disease and their commensals potentially. And then Yersinia pestis might have been already a major cause of pandemics in the past. So we've seen it in the Stone Age. Probably we should really intensely now look also at those earlier historical outbreaks like the Antonine Plague, like the Plague of Athens, other large outbreaks at the end of the Bronze Age in the Eastern Mediterranean. So it's really possible that Yersinia pestis has shaped human history for actually thousands of years, starting in the Stone Age and actually all the way into the 19th century, which I think is quite exciting. So with that, I'd like to acknowledge many people here in the room and other collaboration partners, also my entire department and group at the Max Planck Institute in Llena, a lot of people that funded the research over the last couple of years. And thank you for your attention.
Thank you, Johannes, for a trip through pathogens that's filled with verve and new information, very important new information, very exciting. Let's begin our reaction with some thoughts from my good friend and colleague, Kyle Harper, Senior Vice President and Professor of Classics at the University of Oklahoma.
Great. Is this on? Yes. First of all, thank you, Mike. It's an honor to be a part of this and to be present at such genuinely exciting and really rare, all too rare, meeting of minds across so many kinds of aspects. And thank you, Johannes, for all of your work as a historian. What you do is one of the most exciting things happening in the field, and for presenting such an exciting overview of what we're learning about what I think amounts by any estimate to one of the worst mass murderers in all of history. And I just would start by putting into context a little bit of what you've said from a historian's point of view, how I hear what you're saying, because I think historians should be paying attention to what you're doing, not because it's sort of an interesting solution to a cold case. This isn't an episode of Unsolved Mysteries to identify what caused these pandemics. It intervenes in ways that should really challenge historians to reread the sources, to rethink the way that we understand the role of diseases in history and these great pandemics in particular. And just last year, one of the most eminent medievalists published a synthesis of medieval history that covered very briefly the Justinian Ik Plague, and did so in a way that was entirely dismissive of its significance, and cited a very old and outdated article minimizing the impact of the Justinian Ik Plague and claiming its narrow geographic origins or geographic impact and unknown pathogenic cause. And this was, of course, after the really remarkable papers of the last few years that have already told us what, in fact, was the identity of the pathogen that caused the first pandemic, as we ought to call it, the Plague of Justinian. And so it not only tells us, what your work not only tells us the identity of this bacterium that caused the great pandemics like the Plague of Justinian, that in turn helps, I think, to enrich and change the way we can read the historical sources, and even at times, frankly, validates them. Our two main eyewitness sources to the Plague of Justinian, Procopius and John of Ephesus, offer detailed descriptions of the pathology of the disease, which should have led us all along to be very suspicious of Yersinia, but to have now molecular evidence that validates those historical sources is in itself quite important. But the discovery of the molecular evidence for Yersinia pestis as the agent of the first pandemic is significant in another way too, because of the location of its discovery. I think all along the hardest question in deciding what was the impact of the first pandemic is knowing exactly how widespread its devastation was, how deeply it penetrated, for instance, the Western territories and places like the rural countryside that were far from the cities. No one who's read, I think, Procopius or John of Ephesus has any doubt that this was quite a destructive disease in Constantinople. But now to know that it reached 2 Western villages of a few hundred people, perhaps, helps really in substantial ways to build the case that this disease was in a lot of the dark places on the map where we simply don't have any written evidence. And so I think that the molecular evidence is strongly supporting a maximalist interpretation of the first pandemic. So what you're doing is hugely significant, but I want to offer two kinds of questions. One is, what can the molecular evidence now or perhaps in the future tell us, do you think, about why the disease becomes pandemic? I think personally that there's a strong case to be made that the 6th century pandemic in the age of Justinian is the first true pandemic of the bubonic plague, that it had existed for thousands of years, it had afflicted humans in various ways, but that the plague of Athens and the Antonine plague are very likely caused by other agents. Time will tell, hopefully, with molecular identification, but at present I'd be skeptical that those are likely to be caused by Yersinia pestis. So why then does a disease that may have existed for thousands of years cause what is clearly a disease episode that is on a scale unlike anything that it had caused before, at least in the circum-Mediterranean and Western Eurasia? Are there some combination, do you think, of ecological factors or genetic factors? When you look at the Stone Age plague, you've already mentioned that it lacked the ability to produce the murine toxin that lets it build the blockage and the biofilm in the foregut that makes it such an efficient means of transmission. Do you think that there are evolutionary changes that lie right behind the Justinian Ik Plague that might have caused it to become pandemic at this moment in time? And so what do you think the genetic evidence will tell us about that? And then second set of questions is maybe even a little bit more historical, but I'd be interested what genetic evidence might be able to contribute to our understanding of why does it happen then, when it happens, and why does it end when it ends? And do you think that there is a case to be made for climate factors? Because we're learning that the 530s and 540s, the moment right before the outbreak of the first pandemic, were truly exceptional moments in the entire span of the late Holocene. They're really the scene of violent climate turbulence, and the coincidence seems not incidental to the connection between the climate turbulence and the pandemic. As well, the 1340s also look to be kind of outliers in terms of climate history. And then as well, the end of these pandemics, and I think it was so interesting to hear you really talk about the way the Justinianic line is an extinct lineage, whereas the Black Death lives on. But both the 8th century and then the 18th century are also periods of warming, and certainly the end of the kind of late antique Little Ice Age seems to coincide with the end of the first pandemic. So do you think that that the genetic evidence can help us understand the kind of deeper relationships in Earth systems between pathogen evolution and climate history?
Yes, so that's very good questions, and I'm afraid that genetics can probably not offer any satisfying answers currently on that. What we can say is that we don't see convergent evolution in the pathogen lineages that then lead to the major pandemics. So it's not that we see that suddenly some larger kind of for example, changes happen in terms of the plasmid structure, that there are no new genes popping up. That all happens in kind of the late Stone Age, Bronze Age, but that doesn't happen in the historical time period. So the pathogen, basically, that caused the Justinianic plague, the pathogen that caused the Black Death, the one that you find in the Grand Canyon today, I would think, at least based on what the knowledge, the biological understanding that we currently have of the plague, have the same virulence. They would cause the same disease. If you catapult back the Grand Canyon pathogen in the medieval time, it could potentially cause the Black Death again, and the same would probably be the case for the Justinianic plague. So there's no reason to assume it was a biological change of the pathogen. Of course, environmental changes are very important. As you outlined, there was this major kind of climatic shifts in the 530s, which probably caused starvation, which caused devastation, which probably also, together maybe with earthquakes, as people have suggested, and other catastrophes, had an impact maybe on the rodent population. Either it increased stress on the rodent population, or it caused major actually explosions of the rodent populations that then after, for example, an earthquake, people have suggested causes destruction. They're actually good for the rodent population because they find a lot of food, but eventually it's all eaten up, the population is under stress, diseases can spread within the rodent population, and then they can actually jump into the human population. Because what we always have to keep in mind for Yersinia pestis, it's a rodent-borne pathogen. It's actually a rodent pathogen, so what we really have to understand better is rodent population dynamics and what's actually happening in the rodent population, because that then affects the humans. The humans are just a byproduct of what's actually going on in the rodent populations. And there, I think we have to do way more research in the future. Because very, very little is known even how the pathogens spread within the rodent population, how they can actually spread into the commensal population. Even the kind of reservoir populations in Central Asia are not well researched and well understood, because might be hard to find graduate students that kind of want to study those animals or something. I'm not quite sure, but it's very little research, and there's also, unfortunately, very little genetic data from wild rodent populations in Central Asia, where probably the origin of the disease is, at least what we think currently. So that's very, very difficult. But I think we really have to maybe talk more with ecologists, people that study rodent populations, that could then link kind of climatic changes. I mean, we're again in a time of climatic change, where probably we see certain patterns that might be similar, where we could potentially link those climatic changes to changes then in the rodent population. But I think that is probably very important. And the other thing might just be chance, right? Why did it happen in the mid-14th century? Why did it happen in the 6th century? Maybe it was just chance. Maybe it was just, for whatever reason, this rodent population came into contact with Kaffa in the Crimea, and then was transmitted by the Genoese into the Mediterranean by ships, and then basically spread all over the Mediterranean. It could also be a chance effect. I do think the Stone Age plague genetic evidence that we now have suggests that at least there were larger outbreaks in the past. So this is why I also say about the Antonine plague and the Plague of Athens, that at least we have to consider plague now as a causative agent. I think it's also based on the what Llena and other people write. It's probably not necessarily a plague. It could be a combination of many diseases. But I think we have to now kind of really focus back on what happened in the past, because what we have during the Stone Age is quite amazing, because in the data set, at least that the group in Copenhagen analyzed, about 10% of all the individuals they looked at had plague. That is almost comparable to our Elvang mass grave, right? Where we know everybody died of plague. Because of course, when we look at ancient skeletons, we probably have a 80 to 90% kind of false negative rate, where we basically don't find the pathogen, but it actually has been there. And if you see 10% actually have the pathogen, you can actually assume it was way more common than we think it might have been. So probably it was actually quite common, and that's almost to a kind of epidemic level. But then, of course, what we also have to keep in mind, that's a 1,000-year time period. That is not 5 years like in the Black Death or so. So I think there's a good chance that it was on a pandemic level already early on. And if we have that in the Stone Age, then there's a very good chance it happened also in the later Metal Ages, or even in earlier in antiquity. So I think we really have to keep our eye now into this direction. And another interesting fact that I'm trying to do now with some colleagues is, if you look at the phylogeny, you seem to see often those events, like this event with the Black Death, where you have the star-like phylogeny. You have many, many strains diverging from each other. We also have that here. In fact, there's a bunch more here, there's a bunch more there. You have many things kind of spreading at that time. We should actually look into the tree if you find other events where a lot of things are spreading. And now that we have a good timing of when things happen here, maybe we actually find that something happens in the 5th century BC, which kind of then points us towards the Plague of Athens, or we might actually find something even at the end of the Bronze Age or something, where we see a lot of things diverging. That would be, of course, quite exciting, because then we could actually use the molecular evidence to find the prehistorical or historical events, and trying to find even more evidence based on the modern data that we already have, which would be quite exciting.
On the words biomolecular evidence, perhaps we can invite Professor Benjamin to come offer a few thoughts to spark some discussion before we take a few questions from the audience and move to.
Thank you, Johannes. That was just a terrific. You've really set the mark, I think, for future investigations using ancient pathogen genomics, as you've described it. And I think the impact that your work has is not limited, of course, to historians of the Roman Empire, but more broadly to epidemics of the past. And surely there's no shortage of good questions that come to mind based on your work. I'd like to raise just a few, and I realize that there are probably no short answers, no immediate answers to any of these, so I don't want to put you on the spot with this, but I'd like to, there's one question that I have that I'll raise in just a few moments that I think actually goes beyond the reach of ancient DNA research, and it'd be interesting to get your reaction to that. But I want to begin with one that's closer to home about the work you just presented, and that has to do with CRISPRs. And I think CRISPRs are known to the general public through all of the news about how the impact of CRISPR technology reaches out as a therapeutic revolution, really, in genetic engineering. But I'd like to put that aside and sort of focus back on CRISPRs in their natural setting, which of course is loci in the bacterial genomes, which are a record in short DNA sequences of the sort of the infectious history of the pathogen, of the bacterium itself. So these are short sequences that represent, that derive from bacteriophage or invading plasmids that are pathogenic to the Yersinia bacterium. And these are fossilized in the genome of various strains. And I noted that's in some descriptions of Yersinia crispus, there are some that are commonly pseudotuberculosis, which, as you point out, is the ancient origin of these. So could these be useful in fine-tuning the migrations of Yersinia, the evolution of it, the distribution of it? Why don't I just stop there and get your reaction to that, and then I'll return.
Yeah, I mean, there's very little also I can say about that, because nobody has done that yet. So I think that's very exciting. That is very interesting to actually look at the kind of imprint that you might actually find in the genome of those bacteria. But nobody has kind of looked at that now. Of course, if you look at this family tree and the genetic differences we have here, you can count them on two hands. There are very, very few. So I think what you kind of might find, even if you look at the plasmids, you can't even build this phylogenetic tree on the plasmids. So doing that on the CRISPRs and kind of trying to kind of reconstruct some history goes really deep into the history of this pathogen, maybe kind of how it came out of this pseudotuberculosis cloud, because it's paraphyletic, so it's one tiny branch in the pseudotuberculosis family that is much, much more diverse. So I think that's more kind of addressing than the kind of deeper evolution, basically, of the pathogen, but nothing that would be able to address for the last few thousand years, because there's too little genetic changes in the gene.
I mean, it's hard for me to imagine how Yersinia, which migrates through rats and fleas and humans, wouldn't cast some light on the microevolution and dispersion of. So there's a second class of questions which I think in many ways is broader and also technically more challenging, and that has to do with epidemics due to viruses. You mentioned in your talk several examples of past plagues that have questionable associations with viruses, and I'm thinking mainly about RNA viruses, where we certainly don't expect the RNA to persist in the ancient remains. So how do we approach the question of searching for causes due to RNA viruses? And the thought that you and I actually have discussed once, at least briefly in the past, is can we use the human DNA of the victims and recover the antibody genes of those victims and test them against modern pathogens, Yersinia being. So it seems to me that going to the human DNA libraries from the victims of Yersinia pestis, which you've studied, can those libraries be used to identify and amplify functional IgG libraries that could be tested against modern arrays?
Which is a very intriguing idea. I think the problem currently is that most of our genetic data comes from bones, and specifically it comes now from petrous bones, which is the inner ear, which is actually a form of bone that doesn't change much after kind of birth. So it's very, very stable, and the osteocytes would probably not be the source of the type of genetic information that you are looking for. So it would only be if you would have some sort of, I guess, white blood cells, which you would find maybe inside teeth, maybe inside infections. Potentially one could look into caries, for example, which is some sort of infection where you have a lot of white blood cell activity, maybe also some other lesions on the body. But the problem would be, I could at least see, is that you have a whole group of different types of cells that this DNA actually derives from. And only a few of them actually carry this rearrangements that one would like to study. So because you get the soup of DNA from all kind of different organisms, it would be very, very difficult to reconstruct then this kind of rearranged genetic signatures, which would actually then be useful to reconstruct a past kind of exposure to a certain pathogen. So I probably would think that it would then be even more likely to say find the proteins themselves or something, look for the immunoglobulins or something like that. That is something I think one should try, and I think we have been discussing that in the past, and it's something that I think one could probably do, maybe going from the 19th or 20th century medical collections kind of further back in time, not start with Neanderthals like people tend to do. But yeah, it's
certainly, I think,
exciting.
The going forward with investigating that question depends on the quality of the human DNA and the amount of vascular. So is the petrous bone highly vascularized? We don't know.
No, it's not.
It's not.
No.
Okay, too bad.
Yes. Unfortunately not. That is also why the preservation is so fantastic, because it is actually very dense, it's very inactive bone, in fact. Yeah.
Well, I just think that using your material, where we start out with known victims of YP, would be a favorable one to test this hypothesis. So a final question that's been raised often by infectious disease historians and modern-day immunologists is whether major outbreaks of plague, let's take the Black Death as an example, imposes a bottleneck, a genetic bottleneck on the human population. This was raised, I think, in part of your talk where you discussed this. It seems to me that if the human DNAs of these victims can be looked at, and particularly the MHC locus, which is the main determinant for pathogen recognition and immune response, that this might be a way to actually answer that question, or at least investigate it. Has there been a shift in the MHC pre- and post-outbreak?
No, it is a very important question. It's also one that I addressed at the beginning, this host-pathogen interaction, trying to understand also how the human population reacted, and is there actually immunity? And going back to Carl's question about why did it happen, and why did it happen so kind of strong at the beginning, at the Black Death and the Justinian Ik Plague, maybe even in the Stone Age, there was very high mortality, and then it kind of went down through the centuries, probably because immunity was building up. But is it just the adaptive immune system, or is it actually the inborn immune system, like HLA, for example, or toll-like receptors, or other genes that give you some sort of immunity? And people have been speculating about that a lot. Already more than 10 years ago, people have done some exciting research where they suggest that there are certain mutations, like the CCR5 mutation, that gives you also resistance against HIV, and people have actually suspected that because in high frequency in Europe, it might have been actually also giving you resistance against plague. But then other people, actually Pardis Abeti here, who's also in Harvard now, actually showed that it's actually a very, very old gene. It's not just kind of rose in frequency in the last a few hundred years. So there has been much speculation, but so far we haven't identified any gene that is basically giving you immunity or something that has been under selection due to the plague. HLA would of course be one obvious locus. We have actually started a project some years ago to do exactly that type of question, but back in the day we thought the Black Death was the first pandemic. Because the Justinian Ik Plague didn't really look like a plague for us in this kind of early genomic data sets. Then we had to learn the Justinian Ik Plague was already plague, so all the samples we collected from the pre-Black Death were not useful anymore because the plague had just happened a few hundred years before, so selection would have already been there. We then started to collect material from the 4th century to then learn that it was already there during the Stone Age. So now we would have to go back to the Stone Age and compare a kind of late Stone Age population to later population, but then there's all kind of pathogens that kind of might have been responsible for genetic shifts. And together with David and Ian Matheson, one of his postdocs, we actually had a study just 2 years ago where we showed that there's a whole bunch of immunity-related genes that have been under selection and changed through the last 8,000 years, but which one of that might be related to plague is very difficult to say. So currently we have a project where we're looking into victims of the Black Death, trying to see if they are somehow genetically different to people which sit here in the room, the survivors of the plague. But that is something that is still ongoing, and so far we haven't found any hot candidate inside the HLA and also among the other 400 immunity genes that we're studying, which could actually give some sort of immunity. But we are kind of certainly very, very excited and interested in that type of question.
Thank you very much, Tom and Johannes. Johannes, you're still standing. Could you
kind of
handle a question or two from the audience?
Sure, absolutely, yeah. Absolutely.
Then let us invite questions from the audience.
Human remains. Oh, yeah. With your directly dated human remains, have you tried to calculate the pathogen mutation rate? And if so, is it faster or slower than you would have expected?
So actually this tree, which is a bit of a mock-up tree because it's not the actual tree that comes out of the software, is based on those mutation rates. Otherwise we couldn't basically do the molecular dating here. And actually we have so many calibration points of the molecular clock now inside the tree that we can actually get a pretty good, well, we have a pretty good understanding of the pattern. And actually the mutation rate turned out to be faster than was thought before. So people actually thought that kind of the most recent common ancestor was something like 20 to 30,000 years old. We now show that it's actually just 5,000 years old. There's also things in the tree which I didn't really give names to here, but some of them are called, for example, Antiqua, some of them are called Medievales, some of them are called Orientales, which are all names from the 1950s where people first started to identify those differences in the different bacterial lines based on growing on different media, and they gave them those names based on the 3 pandemics. And that is actually Antiqua here, for example, but there's also an Antiqua here, and there's an Antiqua there, and kind of doesn't make sense at all with the kind of branching pattern that we now have. But that was also kind of showing that this kind of tree has changed quite a lot now that we have those ancient pathogens as calibration points. And that's actually quite useful. If you, for example, look at tuberculosis, people had suggested that tuberculosis is more than 100,000 years old and came with humans out of Africa. And has been big papers in Nature Genetics only 2 years ago. And then when we actually got the ancient tuberculosis genomes, plugged them in the family tree, we could actually see it's only 5,000 years old. It's actually the same authors with us then on this other paper that we then published where we could show, no, it's only 5,000 years old. And it's very useful to have those ancient pathogens because you really can anchor the whole tree in time, which people cannot do based on just studying in vitro bacteria.
I'm going to ask for perhaps one more question because unbeknownst to you, this man is really amazing. Not only is his science amazing and is his delivery in English amazing, but he just got off the plane. It's 1 o'clock in the morning for him, and I think he's doing a hell of a job. One last question, and then we can continue the conversation with a glass. Malcolm Weiner, please.
Thank you, Malcolm Weiner. Johannes, thank you very much. Good to see you again. Professor Harper mentioned that, of course, the Justinian Ik Plague, and especially in Constantinople, is one where we know the number of casualties, and in absolute terms they were very large. But of course, it's only in the Roman Empire that you begin to get populations that, again, equal the population, say, of the Egyptian Empire or the Hittites. And of course, in Crete, where I just sent off to press a paper today, it looks as though the population goes from about 300,000 people at the height of the Minoan Empire down to about 60,000, which is, you know, an 80% fall. So in relative terms, that's very great. Now, of course, Herodotus tells us that the descendants of the ancient Cretans told him that there were 2 terrible plague episodes, one just before the Trojan War and one after. But we know, of course, from all the Hittite texts and the Egyptian texts that they speak endlessly about the horrors of the plague. The Hittites call it the Egyptian plague, but it takes it kills two successive Hittite emperors, and Ramses V is thought to have died of contamination of skeletons. So the question arises whether we didn't have plagues in the Bronze Age that were just as serious. We know from the text we had germ warfare. Each side tried to send plague-infected sheep behind the lines of the others, but it's really very hard to know whether these incidents, even in absolute terms, but certainly in relative terms, were not as destructive as the Justinian Ik Plague and indeed the Black Death.
Yes, no, absolutely, and this is actually exactly what we're trying to address also with this new research center, that we want to try to find out what caused those major kind of cultural turnovers and probably also genetic turnovers at the end of the Bronze Age in the late second millennium. Because, yeah, we have major devastation. We have things happening all through the Eastern Mediterranean, probably the Mediterranean at that time. And there has been much speculation. Was it just the sea people? Was it just people moving around? Or what caused them then to move around in the first place? And was it maybe also diseases that kind of spread during that time? And you could probably ask that for many of those time periods. And in fact, we have some unpublished, very exciting data now also from the Bronze Age, where we actually have genomes now falling on this part of the tree, so things that have actually the flea transmission that cause bubonic plague. So I think we move this entire kind of bubonic story now also into the Bronze Age, and then it's well possible that the Hittite plague or some other plagues that we know from that time period might actually be caused by bubonic plague. So that's something that now everything, all the human remains that we are collecting currently, we are also checking for, in fact, not just bubonic plague, but actually for more than 150 pathogens currently. So we have done a large screening, one of my postdocs, for 3,000 human remains for 150 pathogens. We found about 450 positive hits throughout the last 8,000 years, so all kind of things. So there's a whole kind of new field that is emerging now, especially with this new computational approaches that we can really do some sort of health check. I mean, we have to assume that most people that died in the past didn't die of old age, but in fact of some infectious disease. So it's many that we can. Certainly, I mean, it causes people to be immunocompromised to some extent if they're kind of starving, and I mean that's what we know from many kind of famines in the last 200 years, and that's certainly a very, very important point as well. At that time period, that pathogens might have a greater chance to spread, like we discussed before, the 530s, or also in the 14th century before the Black Death, there were major famines in Europe. Misharvest for several years, especially at the beginning of the 14th century, which probably have their toll on the population and might make it then easier also for those pathogens to spread.
I propose that we give Professor Krause a resounding round of applause.